Cell theory. How humans understand nature is often because of the instruments that were developed at the time. The development of microscopes provides a clear window to the world of cells. Light microscopes, like the kind used in your classroom, were first developed and used by scientists around 1600. In a light microscope, visible light passes through the object like a thin slice of muscle tissue. Glass lenses increase the image and project it into the human eye or a camera. In 1665, an English scientist named Robert Hooke used a light microscope and saw rectangles when looking at a thin slice of cork or oak bark. He named the rectangles cells. He was looking at the walls of dead plant cells. Many more observations by the other scientists were needed to understand the importance of Hooke's finding. By 1700, a Dutch scientist, Anton van Leeuwenhoek, developed a light microscope with high-quality lenses. This microscope allowed him to see tiny living creatures, such as those in pond water. He described what he saw as animalcules. For the next 2 centuries, scientists using microscopes found cells in every creature they observed. By the mid-1800s, this evidence led to the cell theory. The cell theory states that all living things are made of cells. Those cells are the basic unit of structure and functions in living things. Later, the cell theory was extended to include the concept that all cells come from cells that already exist. The finding of the cell has made a huge effect on how we all live today. Scientists can now treat diseases and cancer because of their knowledge of the cell.